All right, good morning, everyone. Thanks for being here, and thanks for being here early. So maybe I'll just start with just talk a little bit about myself, where I come from. I guess my background is in electrical engineering and mechanical engineering, and I've done a lot of stuff on medical devices. Maybe just to give you a sense of the type of engineer I am, because there are lots of different types of engineers, there are lots of different types of clinicians. I'm a sort of practical design and build type of guy. So a lot of stuff I've done here are like these are like some autonomous underwater vehicle work, so a lot of medical devices work, microfabrication sensors. So I basically I make and build stuff. And lately my research focus is on medical devices, or sorry, microfluidic devices for cell separation and cell valve mechanics. So what I'm going to try to do here this morning is talk about three things. Okay. One, I'm going to talk about microfluidics as a technology, sort of give you some background on how they work, where it comes from, and what it means for life sciences. Then I'm going to review the sort of state of the art in circulating tumor cell separation and its challenges. And then finally, I will present some of the work that we're doing on CTC separation based on cellular deformability. Okay. So the first part is microfluidics. This technology has sort of gotten a lot of attention lately, especially in the life sciences. So I just want to give you a background on it, sort of give you a sense of what it can do and what it cannot do. Okay. So microfluidics originally, I guess the story starts from the first transistor. So that was late 40s that was invented. Okay, and that's what it looked like when it was originally, it's kind of like this sort of clothes hanger type of gadget. And what a transistor is basically a bunch of thin films of different materials put together. Okay, and if you have one transistor, you really can't do much with it. Okay, so pretty quickly, once we had the first transistor, we wanted to make lots more transistors. Okay, we wanted to go from this guy to basically what is the state of the art today, which is the Intel Pentium chip. And to do that, we developed this thing called integrated circuit microfabrication. That was still starting in the 60s, and then the Nobel Prize for that was given out about 5 years ago. Okay, and this technology really advanced quite rapidly. So we went from one transistors to what we can do today, which is a billion transistors. Okay, in a very small area. And the characteristics of this technology is that, well, one is you can get virtually limitless complexity. Okay, in a very, very small area, you can create tons of little patterns. You can create structures that are sort of on the length scale less than 1 micrometer. And that's interesting from a biological point of view, in that you're now approaching the size of biological cells. Okay. But the other part of this technology is very expensive. Okay. And expensive, we don't mean like a Vancouver condo type of expensive. This is like the GDP of a small country kind of expensive. Okay, so the last Intel fab that was built in 2005 that cost $3 billion. The latest fab, TSMC, which is a Taiwan semiconductor company, the biggest manufacturing company in the world for microchips, it cost almost $10 billion. Okay, and so as you might have noticed, computers hasn't really gotten cheaper in the last few years. You're kind of really hitting up against a cost limitation here, right? So this is very expensive technology, great technology, but it's very, very expensive. So the second part of this, of the origin of microfluidics, is this idea called Micro-Electro-Mechanical Systems, or MEMS. So once we had the capabilities to create complex patterns at the small length scale, people started wondering, at least on the research side, can we create devices that have both electrical and mechanical features? And this has been talked about for a long time on the research side, but really it did not get a lot of traction. Okay, there are sort of a few niche products. So originally there was accelerometers for cars for deploying airbags, and projector digital. A lot of projectors are made using these sort of arrays of micromirrors. And actually the thing that really saved this whole technology, the MEMS technology, is smartphones. Okay, now, because every smartphone has an accelerometer in it now that tells you which side is up and whether you're moving or not. Okay, and so once again, the characteristics of MEMS, which it's basically a cousin of the original integrated circuit microfabrication, is it's a high value item. Okay, it's not not really disposable. Okay, which is not good for biological processes. And it's got a really long development time because this it's something like this is very difficult to create. So typically product that would take like 15 years to develop, and that's what they took for the original accelerometer as well as the DLP projector. Okay. The other thing is that you can create these complex structures like this. Okay, these are like gears that are extremely small, like sort of at the length of the diameter of the gear here is about the same diameter of a human hair. This is sort of it's a planar process, so it's very difficult to encapsulate this. Okay, so if you're trying to work with biological samples and working with fluids, ideally you want something that's fairly encapsulated. Okay, so this is mostly a laboratory curiosity for a long time. Okay, and but then about 15 years or so ago, people started thinking, okay, can we address some of these issues, and can we start to use some of this technology as a starting point to make things for handling biological fluids? Okay, and that came about the idea of soft lithography. And what soft lithography is actually it's a really simple implementation of a very old technology called molding. Right? So first you create the structure. Okay, you create microstructures like this. Okay, whichever method, but let's say you integrate circuit fabrication to create a structure. Then you pour like a soft silicone over this structure. Okay, you let the silicone cure, and it sort of hardens into this gel. Now you can peel the silicone off, and you can now bond it to something else. And so what you've done now is you replicated this original structure. You made an inverse copy of the original structure, and this thing is now you have an encapsulated device. And now if you poke some holes in this thing and put tubes into it, okay, now you can have this very small area to process fluids. Okay. And so this was a way to make to create microstructures and to make it cheap. Next, if you want to really work with fluids, you want to have some way to control this. Then there's this idea of multi-layer soft lithography, where you take structures like this, except now you can create multiple copies, and you can stack them on top of one another. And when you stack them on top of one another, the intersection points is you get a thin membrane, and if you apply pressure to one side of the membrane, that inflates the membrane. Okay, and that membrane then bulges up, and when it bulges up, then you can use it to seal off a channel. So now not only do you have microchannels, you can have valves that sort of control the flow of the fluids. And I don't want to go too much into this, but just sort of starting to summarize here. So what you get is, starting with integrated circuit fabrication, you can get microscale structures, you can get complex patterns, and with molding, you can get encapsulated microchannels and the economical means of fabrication, which is very, very important. And then finally, with microvalves, you can get flow control and automation. And so what does this really mean for life sciences? Well, these capabilities give you sort of two things you can do. Okay, one is you can work with small volumes of samples. Okay, so if you work with small volume samples, you minimize costs, you can potentially preserve signal, and this is good for like large multiplex arrays. So if you're doing combinatorial chemistry, if you're doing drug screening, if you're doing genomics, where you want to have lots and lots of reactions, hundreds and thousands of reactions, millions of reactions even, that you need to do on a single sample, this is really good. Okay, and most of the world, is sort of focused on this small volume idea. And another thing is that another idea that's sort of come in vogue recently is that this idea of being able to create microenvironments. So you can create simulated organs and tissues and organs which you can apply therapies to, and you can kind of see what happens. And that's something that's starting to come online. Okay, and then on the small length scale side, you can perform precision flow control. You can do some interesting fluid physics. I know it doesn't mean much right now, but and then there's sort of structures. The other thing is you can create structures or at the length scale of individual cells, and this is where really we're going to focus on in our work. So I'm going to try to switch gears for a second and talk about CTCs and sort of the state of the art of CTCs, and then I'll come back to applying some of these technologies, microfluidics, to the problem of separation of circulating tumor cells. Okay. Okay, so I don't know how much you guys know about circulating tumor cells, but I mean, based on, I guess I'll just try to start from the beginning. So, based on the seed and soil hypothesis, you have a tumor, and that tumor metastasizes. One of the things that has to happen is that you actually have to have individual cells that are detached from the tumor that are transported through the circulatory system that then is then seeded somewhere else. Okay, and the current idea is that if you can have some way of capturing these cells that are in transit, if you can have some way of characterizing these cells, then you can create a blood-based biomarker for metastasis. And the implications of that would be enormous in terms for therapy. I mean, the list would go on and on, but basically you can think about different ways to do tumor staging. You can look at it as a fluid biopsy of the original tumor of a particular invasive population of the original tumor. It's a potentially a real-time biomarker of disease status, and for these folks doing drug discovery, it's potentially it gives you a very fast marker for drug efficacy. So instead of looking at survival rates and things like that takes a very long time to gather. Potentially this type of marker can tell you whether a drug is working right away. Okay, and then also because it's a blood-based biomarker, there's no reason why you can't just take more samples at more frequency, so you can look at the long-term surveillance of indolent disease. So the whole, the field itself is just really exploded in the last 5 years because all of the potential things you could do once you had access to these samples. So this is sort of the promise of CTCs. Now the problem of CTCs is that they're extremely rare. Okay, and so the estimates are typically around sort of 1 in about 5 million leukocytes. Okay, and leukocytes being the primary contaminant. So red blood cells are almost inconsequential in this. It's mainly CTC. How do you distinguish CTCs from leukocytes? And so what's being used as has been established as this idea of existence of CTCs as it's associated with poor clinical outcome is that really we're looking at like a very, very small number. So what we're talking about greater than or equal to 5 cells per 7.5 milliliters of whole blood. Okay, and 7.5 milliliters of whole blood, there will be about 40 million cells or 40 million leukocytes in that sample. Okay, so this is so these are based on extremely small numbers, and everyone's looking at this saying 5 cells. Really, is it really going to be how credible is that measurement? And the issue is that if you increase this number to a bigger number, say like you have to have more than 30 CTCs, or you have to have more than 50. Well, then you're going to have a very small number of patients that actually would fit that criteria. Okay. So to be as inclusive as possible, okay, 5 is the sort of the current number. Okay, and there's been all these different survival curves done to show that okay, indeed if you have more than 5 CTCs, then for the prostate, breast, I think, and also colorectal cancer, that there is definitely poor outcome associated with CTC. And so to talk about the different ways to separate CTCs, let's just look at the overall picture conceptually. Okay, when you have something as rare as 1 in 5 million, typically a one-step process is just not going to cut it. Okay, because every process has its own errors and its own biases. So generally speaking, you first have some kind of enrichment step that would go from sort of 1 in 5 million, okay, down to the 1 in 1,000 to 1 in 100, which is still pretty rare. Okay. And then you would use some method to sort of identify the CTCs. So generally speaking, you use immunofluorescence of cytokeratin. So what you believe is the tumor cells will have cytokeratin and the leukocytes will not, and then the leukocytes will be positive for CD45, and the tumor cells would generally not have CD45. Okay, and so you have some way to, so you would be reviewing all of these cells and then trying to figure out, okay, a small number of that fits this criteria, being CK positive and CD45 negative. And then the next step that isn't really currently being done much in the field is, looking at single cell characterization of a small number of cells. And there's a whole, plethora of biological characterization you can do if you had the cells. And so the real issue here is